At first glance, this scientific poster looks complicated. However, it's just a result of my personal bioinformatics research project from 11th grade. Bioinformatics is a new emerging sector of biomedical engineering and will be an incredibly useful tool in future research, according to Taylor from the Science magazine in 2000. But what exactly is the definition of bioinformatics? According to an NCBI article by Luscombe and colleagues in 2001, bioinformatics is a process to solve biological problems through computer science, chemistry and biochemistry, applied mathematics, and statistics. In essence, bioinformatics is research conducted using a database of genomes and genetic information. This video will work with the IMG database, short for Integrated Microbial Genomes and Microbiomes, according to their website. By the end of this video, you will be able to create datasets in the IMG database to help you start your bioinformatics project. You will understand what you need to get started, the process itself, and things to avoid during the process. Before you begin, you will need a computer, lab notebook and writing utensil, an IMG database account, as this is the database I will be using in this video. Different databases work differently, so make sure you are using an IMG database account or the steps may not apply. You will also need a strong and reliable internet connection and the research project that you hope to complete. The first step is pulling up the IMG website. From here, select Login in the top right corner. Put in your password and login information and select Sign In. To begin, know that your dataset will be the collection of genomes you are working with. As a reminder, a genome is a collection of all the DNA an organism has, according to a 2018 article by the U.S. National Library of Medicine. To find genomes to work with, select the Find Genomes tab from the toolbar and scroll down to Genome Browser. The database will load as seen here. The amount of data the program needs to sort through is very large, so the program will take a long time to load. This is why strong internet connection is so important. Do not refresh or exit out of the page while it is loading, or you will have to start over. This becomes incredibly important later when you have unsaved data sets. Once the database does load, select View Alphabetically and let it load again. It will load the same way, and a pop-up block will also appear saying Retrieving Data. This is just another form of loading. Let the program load, do not refresh or exit out, and wait for it to produce a page with a table on it. The database will produce hundreds of pages of a table with multiple columns. Let's discuss these columns before getting into how to find genomes specific to what you want. The first column, Select, is just as it says. When you decide you want to use a genome, simply click the button and you will be able to add it to your database later. The second column tells you the domain of the sample, archaea, eukarya, or bacteria. Next, the sequencing status lets you know if the DNA is completely sequenced, denoted by finished, or only a draft. The study name column gives a brief description of the sample. Information here might tell you why the sample was collected, what study it was sequenced for, or even what the organism is in non-scientific terms. Following this, the genome sample name column gives you the specific scientific name of the sample. The sequencing center lists where the DNA was sequenced, and the last important column for our purposes here is the IMG genome ID. As you select samples, write these IDs down in your lab journal. It is easiest to search specific samples back up by using this ID in the database. If your data is lost by an internet crash or refreshing the page, and you have written these numbers down, it is easy to recover your data. If not, it may be harder and more time-consuming. Knowing how these columns work, you can decide how to look for what samples you would like. Select Filter column and choose which column you would like to search under. Here we'll use genome or sample name. Then, search for what you're looking for. Make sure your search is specific to the column. If it isn't, choose the appropriate column. Let's search for the bacteria Listeria. Clicking the Apply button, the system will load and bring up the results. Our results will have all the genome sample names containing Listeria. To select the datasets we want to work with for the duration of our research, we'll select those we like best. You can decide which work best for you based off the information in any of the columns. Note that you can change pages to see more data by the numbers here. Now that we've selected those we like, we'll scroll to the end of the table and select Add Selected to Genome Cart. From here, we'll be brought to the genome cart with the sets we selected. These sets will not be saved if we log out of the program and log back in. To have them when we log back in, we need to save them to the workspace by selecting all the data sets, clicking Upload, Export, and Save, and scrolling to Save Genomes to My Workspace. From here, we save them under our desired name of the set and click Save Selected to Workspace. These can be accessed later by going to the workspace in the toolbar, selecting Genome Sets, and seeing which of the sets we'd like to use. We can also select them when we're logged in without going back to the workspace in this toolbar here. Hopefully, this video has taught you how to start bioinformatics research in the IMG database, from what supplies you'll need to the process of creating datasets and what things to make sure you avoid. In no time, you will be able to create presentations like the one you saw at the beginning.